All right, so you guys just talked about how plyometric training can increase the stiffness of the overall tendon and improve performance. I'm going to talk about a little bit of the opposite. So when you're loading on a tendinopathic tissue, so not a healthy tissue. And we think tendinopathy develops out of an acute injury to this tendon matrix here that disrupts the alignment of the collagen fibers. And this disruption of collagen fiber alignment and scar tissue formation is really important when we start to consider how we're placing load on the tissue. So in a spectrum of the way to load a tendon, you can either load it really short and fast, and we're showing that here with the peak really high deformation, the loads being elongated, the tissues being elongated super fast in a short amount of time, whereas a different loading pattern here with the tendon being elongated and held at that tension. And when this type of load, this short dynamic load, is applied to a tissue, the load can pass through these strong collagen fibers and avoid this injured matrix within the center. And that's something we refer to as stress shielding, where the stress that's applied within the tissue, it is shielded from this injured area here. And it gets shielded from this area because the injured region of the tendon is weaker than the surrounding part. Whereas during something like stress relaxation, the tendon is elongated, held at a certain length, and eventually once these strong collagen fibers start to relax, load can pass through the injured region of the tissue. And I have a little demonstration for you. So this here is a tendon. You can see all of its nice long and aligned collagen fibers. When I load the tendon really quickly, nothing happens to it. And that's interesting. It's kind of cool because the load starts to pass through all the strong collagen fibers in the paper, just like the tendon. But when I hold here, I'm not pulling any harder than before. Eventually, the strong collagen in the tendon, or in this case a piece of paper, starts to relax, load passes through the center, which was the torn piece of paper, and eventually the tendon ruptures. So to look at how gene expression changes within a tendinopathic tissue, we did an experiment where we took injured animals, we injured the patellar tendon on one side, punched a hole straight through the center, and let that tissue scar over for 2 weeks. And then after 2 weeks, these tendons were either loaded isometrically or dynamically. And what's important is that these isometric and dynamic loads have the same amount of tension on the tissue, but this isometric loading on the patellar tendinopathy was 4 sets of 30-second contractions, where the tendon was held at a certain length for 30 seconds, a really long time, whereas during the dynamic load, the tendon was loaded short and quickly, 360 times for a second on, second off, over and over and over again. So when we did that loading pattern 2 weeks after the injury and collected the tissues a day later and looked at changes in gene expression, we saw changes in gene expression as a result of the loading. And most importantly, we saw during that isometric load, that long load where we think the tendon starts to undergo stress relaxation, we saw a marker called scleraxis, tend to increase in the isometrically loaded samples. And it actually statistically increased when we just look about the amount of scleraxis in that tissue. So what is scleraxis? Why is it important? It sounds weird. So scleraxis along with mohawk, these are both tendon-associated transcription factors that are important in promoting a tendon-like phenotype. And what you can see is that when we compare expression levels of scleraxis in the injured and loaded right leg relative to the left leg, it starts to increase. And when we just look at the absolute levels of scleraxis in the injured and loaded right leg, it actually shows a statistically significant increase. And we saw a similar trend in mohawk, which is another tendon-associated transcription factor, where levels in the right leg relative to the uninjured left leg started to increase, and as well as with tenomodulin and fibromodulin. So tenomodulins, a glycoprotein on the surface of mature tendon cells that marks that these cells have a mature tendon cell phenotype, whereas fibromodulin, it's a small proteoglycan within the tendon matrix that's important in organizing the collagen fibers. And what I should emphasize here is this entire top row, these are comparing expression levels of these genes in the leg that was injured and loaded relative to the leg that was not injured or loaded, the control leg. Whereas expression levels here are just the absolute expression levels of these genes, not relative to the other leg. So when we go on to look at some of the structural proteins that are present within the tissue, so the collagens, these again showed a similar trend where collagen type 1, the main long strong protein within the tendon, tended to increase in the isometrically loaded samples. And again, this is a huge fold increase, over 2,000 fold in one sample. Whereas collagen type 2, so again, it's another structural protein, but this type of collagen, it's a little different. It's not a long, strong protein. It forms more of like a mesh work. And that mesh is usually present in cartilage. And we saw that the type 2 collagen started to go up in the dynamically loaded samples. And this is really important because it's suggesting that that short, fast dynamic load on the tissue is squeezing the cells so hard that they start to turn into a collagen or a cartilage phenotype. And lastly, we looked at type 3 collagen, and not surprisingly, type 3 collagen can be indicative of an injury to a tissue, and it is, again, increased in all the samples and tends to increase a little more in the isometrically loaded sample. So to summarize, training with plyometric loads, so such as sprinting, jumping, really short transitions that are present in like basketball or volleyball, can result in stress shielding such that the injured tendon never sees the load. So that's the injured spot, that scar tissue within the tendon. The stress gets shielded around it and that area never sees it. It never sees a tensile load, and that tensile load is needed to deposit collagen along the line of force. Whereas training with isometrics or heavy, slow, concentric or eccentric moves can result in stress relaxation through the strong portion of the tendon and ends up sending load, directional load, through the injured region. And that directional load is important because all the cells within the tendon, all the tenocytes, need a tensile load to make more collagen and put it down in an aligned manner. So, and I showed you in this study that isometric loading of an injured tendon can increase the expression of the tendon-specific marker or transcription factor scleraxis, and it tends to increase the expression of other tendon-specific genes, such as MOHAC, another transcription factor, or fibromodulin and tenomodulin. In contrast, we saw that dynamic loading increases the expression of the cartilage-enriched gene, collagen type 2, and that type 2 collagen helps resist compression within cartilage or within that injured region of the tendon. So this is suggesting that the isometric loading could reverse tendinopathy by getting a directional load through the tissue and increasing the expression of genes that are important in maintaining a tendon phenotype.